The same religion that's capable of hideous acts of destruction can also be capable of moments of healing, of restoration, and of hope.
But educate a girl, and you educate her entire family.
There is a sun within every person.
When that anger sets in, write it. Write the letters, but don't send them. You never want to leave concrete proof of insanity.
Speak to you about systems biology, and I picked a specific example to motivate this, to kind of galvanize or focus things, and that's circadian rhythms, something that I know all the travelers in the audience have a first-hand experience of. Let me first start by pointing out that we have a very richly interdisciplinary team here. I think you're going to hear that motif time and again today. Chem engineers, biomolecular scientists, computer scientists, electrical engineers, all working on the research team to try to get at what are the design principles that nature uses. I'm a control engineer by training, and so we work with biologists at various institutes, as well as collaborators here in the computational science side, to try to unravel the principles in these problems. And our funding has come largely from Department of Defense, the Air Force, the Army, and DARPA. So this work that I'm going to describe to you, I'm going to first back up and give you the big picture here on the campus of systems biology. And a lot of this is happening within the Institute for Collaborative Biotechnologies, which you heard a little bit about yesterday from David Gay. We have a vision in the ICB that network science as a discipline is, I think, coming into a focus, coming into definition right now. And the kinds of problems that we see in biology, this is the architecture that underlies the regulation of a key gene here that controls one particular aspect of a very simple organism, C. elegans, a worm, at one point in time, and you have all of this massive redundancy and regulation to control that one protein. We think that by unraveling these kinds of networks, we can begin to get insights into more complex engineered networks, like the kinds of communication networks problems that our sponsor, in this case the Army, is very interested in. So we're looking for those core technical challenges at the interface between biology and engineering. So I want to start first by giving you a definition of systems biology. This is one of these buzzwords that probably means many things to many people. I was fortunate to participate in an international benchmark study that sought to define and identify the opportunities in this field, and we came up with the following definition. I've highlighted the word in red specifically for the discussion today, and that is that the network attributes of the system really are what distinguish systems biology from more classical reductionist thinking and the kind of thought that dominated biology for the last 2 or 3 decades. I would argue there's effectively a three-legged stool in this problem in systems biology. We need to have good biological problems, we need to have good technologies for probing the problems, and we need the side that I come from, the computation and systems side, to model, to analyze, to interpret the data. And I think the rationale hopefully is very clear to everybody. We're past the point of thinking that a single gene is going to explain in a profound way a disease state. We recognize that something as complex as a disease state is, again, the response of a complex network. If you will, the symphony of interactions that occur that give rise to either a healthy state or a disease state have to be understood in the context of the network. We're well past the point of thinking that just the components will tell the whole story. And some of the understandings that have emerged, particularly from the community I come from, the control and systems community, is that nature, much like engineering control systems, is very hierarchical, strongly layered. And there's this overarching theme that you'll see a couple times in my talk here, that natural systems, much like engineered systems, achieve robustness at the price of fragilities in some parts of the system. Now, part of the paradigm here in combining the data with the modeling is an iterative motif. And this is nicely articulated by Trey Eidecker and Doug Laufenberger as the quote-unquote 4 M's of systems biology. So you could start in any particular box here, but think about collecting the data, let's say by manipulating your experiments. Collecting that, this is the technology piece I referred to before, so the measure M. That can be mined, because typically these are high throughput techniques, so you're going to generate massive amounts of data, archive that, annotate that in databases, mine the databases, abstract information from data, and use that information to drive the generation of new mathematical models. Then those models drive the next generation of experiments, and you continue going around the circle here. And you go until you stop at the point of either validating a hypothesis, increasing understanding, or if you're in, let's say, the pharmaceutical sector, identifying targets or therapeutics would be a convergence point for this iteration. So that's one of the overarching paradigms here. Ours is a campus where lots of exciting things are happening in systems biology. I'm giving you just a sketch, and for my colleagues in the audience, if I missed your keyword here, I apologize. But in the systems domain, we have lots of folks in engineering and computer science working on different aspects of analysis and modeling. On the technology side, in fact, there's a competing talk that Tom So's given right now describing some very exciting developments in the technology arena, as well as others in biology and engineering here. And a whole host of problems, looking at everything from prokaryotic systems, bacterial heat shock, for example, through the whole range of eukaryotic systems from yeast up through plants, simple animals, and ultimately human scale problems. So a very exciting time at UCSB to be working on systems biology. This has been enhanced by a very generous gift that one of our retiring faculty members gave just a couple years ago. Duncan Mellichamp and his wife Suzanne have endowed 4 professorships in the field of systems biology. We have an offer out to the first potential chairholder. We're hoping to converge this quarter and secure that candidate and hire 3 more, hopefully in the next cycle. The other thing we've done recently is last fall, we hosted the first international conference on the foundations of systems biology and engineering, FOSB for short, with a lot of attention from both corporate sponsors as well as federal funding agencies. So we think there is a certain focus on this campus in this very exciting new area. So let me turn to the problem that we've spent about the last 5 or 6 years working on, and that is bringing these kinds of methods, these kinds of tools, to understanding the basis or the design principles of the generation of circadian rhythm. So circadian rhythm is a phenomena that dominates the animal kingdom, in fact, all the way down to cyanobacteria, keep time with cues from the sun to regulate a 24-hour clock in your body. So those of you that travel a lot and have experienced jet lag, it's because you take your clock, which is very robust, and you shift it by phase, let's say over to Asia, where I was last week, and you've got an 8-hour delay to try to cope with, right? And so the clock is remarkably adept, whether it's in a plant, this is a Arabidopsis, this is in a fly, Drosophila, or in a mouse. You have a very rich architecture, which if you drill down to a molecular scale, is really a sequence of nested feedback loops. So I'm showing you here, these are the 3 key genes in a mammalian system that are responsible for keeping that timing, keeping that 24-hour rhythm. They produce their corresponding proteins, which go through different activity modulations, phosphorylation typically, and feed back and ultimately repress the activity of their genes. So there's a great big time-delayed negative feedback loop here. In addition, there's some positive feedback loops that play into this as well. So as an engineer, I'm struck by this sort of pathway diagram as being comprised of the usual circuit components. Positive feedback, negative feedback. We've got time delays in the form of activity modulation of proteins, gain modulation through the phosphorylation state of the proteins, and indeed a richly, as you can tell from this cartoon, a richly hierarchical architecture that nature is using effectively to keep 24-hour rhythms, to keep time. Now, one of the sort of tenets of systems biology is that if we want to understand how the organism responds at the top of the scale that I've drawn here, we have to do something more than just collect the sum of the parts, right? The usual cliche. But the idea here is that we can't just drill down and hope to explain on the basis of genetic understanding how the clock works, but we have to build back up. We have to integrate across the scales. So I'm going to tell you about efforts that we've been doing that reach up to roughly the cell to organ level, and maybe give you some a teaser about thoughts we have on getting all the way up the hierarchy to the organism scale. So let me talk about the network level, which is sort of an intermediate level in this picture here. And I just want to give you a kind of broad brushstrokes on how we're attacking this problem. So if you dig into the brain, and the hypothalamus in particular, there's a region called the suprachiasmatic nucleus, SCN, where the neurons live that do this timekeeping. There's about 10,000 of them, so this cartoon is not to scale. 10,000 neurons, the ensemble of which, or the collection of which, are responsible for generating that 24-hour rhythm. And that rhythm then drives other organs in your body, but the core clock is basically that piece of tissue in your brain. And one of the remarkable things that's come out of the literature just in the last 2 years is the insight that those individual neurons are crummy timekeepers. If we look at inside an animal here in vivo in a mouse, the generation of period, we see a very crisp timekeeping. It looks quite good, right around 24 plus or minus a small fraction. If we look at an individual neuron and track it cycle to cycle, it's reliable. It's keeping the same rhythm cycle to cycle. If we take a slice out of the animal, so it's still an intact tissue, so-called explant, we begin to see a little bit of degradation in the period timekeeping. It's not quite as crisp on 24, and cycle to cycle, each neuron has more variability, maybe a couple hours plus or minus each beat to beat of the neuron. Now, suppose I do a very diffuse culture where I can actually separate the neurons and keep track of an individual neuron. Now you see where my title here, that the individual neuron is a terrible clock. If I track an individual neuron, it's got a huge amount of variance in terms of the period, especially cycle to cycle. The same neuron may change by as much as 10 hours from today to tomorrow. So I've got components that have these kind of performance attributes. And yet the ensemble of the components gives rise to a remarkable performance. How is this possible? Well, I don't want to get too much into the biology, but there is data that suggests that there's a linking agent. And for those of you that have a biology background here, what we're doing is creating mutants of a putative receptor, the channel that we can use for talking between the neurons, a putative agent, the signal that's being sent in between the neurons, and we can compare that to wild type. And if I disrupt the channel or if I disrupt the agent, I can break down the rhythms. So we think we have a good hypothesis for the means or the mechanism of doing the communication. And we can transform this into a very simple mathematical model. This is that very complex picture I showed you on one of my first slides of the circuit, the core clock at the cellular level. And now we just allow the cells to communicate with other cells. So we put in the agent that would represent that signaling component and the channel by which that signaling component speaks to other neurons, and we let that drive the clock, influence the clock, and the clock sends an output, which is the agent now that can communicate with other neurons. And we can put these on a grid. The corresponding mathematics aren't as important here, but we can put these on a grid and now create an array of neurons. This tissue in the brain is effectively a flat tissue, so we just need a 2D array of neurons that communicate across the grid, and we can begin to try to capture that principle that we see evidenced in the data. And to cut to the chase, we can do that. So we can take a population that are singing in harmony, if you will, here, that have a very crisp, this is now a computer simulation, crisp timekeeping. The organism we're simulating here actually has a slightly larger than 24-hour rhythm, but a very crisp timekeeping. And if I simulate the blocking of the channel by which communication occurs, I get the kind of data like we've seen in the mouse data I showed you earlier. So we can create both the synchrony as well as the asynchrony corresponding to lack of communication in these channels. We can go on and do more detailed analysis of this network. So for example, we can use tools from engineering, so-called sensitivity analysis tools, to now describe, in this case, an ensemble of 120 neurons that can have up to communications between 10 or 12 other neurons. We can predict what are the best coupling agents between the different neurons, putative channels, putative agents that can communicate. I've told you about one, but there are many other hypotheses in the literature. More important, we can make predictions. Suppose I said, I want to perturb components of this lattice architecture and predict how the period would change. If we do that, we can create the following kind of diagram where this would be the actual change in period. This is what our analysis would predict, and you get a reasonably tight correspondence here with an R squared of 0.74. I want to reiterate, this is on the basis of a very simple single deterministic cell, and the data I'm generating is from a large collection of stochastic cells. So actually that R squared value is remarkably predictive. The other kinds of things we can do is we can explore the impact of so-called heterogeneity. This ensemble of 10,000 neurons are remarkably diverse. Some of them are strong oscillators, some of them are damped oscillators, some of them need a drive to oscillate. And we can simulate the increasing impact of adding noise in the system. The more noise we add, the measure of harmony, the so-called synchronization index, degrades to virtually asynchronous, which would correspond to this condition where I have a very diverse spread of periods. For very low noise in the top plot, I get my tight, crisp synchrony. So we begin to get a sense of the trade-off not only within the cell, but the properties of communication at the network level, of how the trade-offs between cell-specific attributes and network attributes give rise to the kind of performance I showed you over there. Okay, the final thing I want to show you is a more dramatic demonstration of this phenomena. Everything I've shown you so far is just about a network, the neurons, and how period might be adjusted for small perturbations. Let's make a radical perturbation in the system. Let's introduce a phenotype by doing a mutation on the organism. And in this case, I'll show you some data from the literature. This knowledge is probably 10 or 20 years old, but there's a good study in '99 that showed if you knock out critical genes in that architecture, you can still keep rhythms. In this case, they're measuring the activity of a mouse running on a wheel that has the genetic mutation of knocking, let's say, the Cry1 gene out, or the Cry2 gene, which says that, in effect, these are dispensable components. I don't need that component to get rhythms. I might change the period of the rhythms, but I can still get rhythms out of that organism. Now, what's exciting about this is this was done at the mouse level. This is the organism scale in my hierarchy. Now, within the last 6 months, people have begun to do experiments at the single cell level. They can manipulate individual single cells, and now what you see, if you do a Cry1 mutant, in the mouse, you got rhythms, the period changed, but in the single cell, you lose rhythmicity. It's so-called sputtering phenomena, where occasionally you'll get a spike and then it'll go silent for a couple periods. So the phenotype, the behavior at the single cell level, is fundamentally different than the behavior at the network scale. Components versus network, completely different story. So any mathematical model or analysis I do here that presupposes knowledge of the cell is wrong, because the cellular behavior contradicts the tissue behavior. So we can put this in the framework that I described to you earlier. We can take a core oscillator model, like the kind we talked about before. I can do a slightly more detailed mathematical formulation. In this case, I will break all of my chemical reactions down into so-called elementary steps, and I'll have them fire at stochastic intervals. This is much more consistent with the molecular biology. And then I can put together an ensemble, and so this cartoon is meant to represent a number of those core oscillators arranged in an array, and the pink kind of cloud here denotes the diffusion of that peptide that's communicating between the oscillators, so I can begin to simulate with a coupling rule the mutant phenotypes and see if I can introduce the same kind of behavior that we saw in the experimental data. And so here's an example of the actual experimental data in the intact network. So this would be like the mouse running on the wheel. You get very nice rhythms. This is a raster plot of about 100 neurons over about a week's worth of data. Nice rhythms. When you look at the individual cells, rhythms are gone. That was the dramatic phenotype I talked about before. Our simulation results, nice rhythms for the network. And indeed, we can replicate the idea that you get asynchrony in the population and these so-called sputtering neurons for the simulation model to validate this. Okay, so let me kind of wrap up here. What I, in a very short period of time, hope I can convince you of is that this problem in systems biology that requires integration across scales really requires thinking about performance and robustness in a different way than engineers usually think about it. For example, the questions I might pose at the gene scale or the protein scale, the network scale, all are different. The attributes change. I tried to convince you that with the simple analysis of cells versus combinations or tissue level analysis of that performance. So context is key in biology. We need to be very clear about what scale we're analyzing. And more important, we think this can generate insights for pharma applications. For example, novel targets of the clock to manipulate the clock. And so the last thing I'll leave you with is some exciting new work we've just started doing in which we've taken control engineering ideas, the way that we control things like petroleum refineries, so-called model predictive control, and thought about applying that to the clock. How would I push nature's natural timer to reset it more quickly? Suppose I was out of phase 8 hours because I flew to Japan. How do I reset the clock without waiting the usual 6 or 7 days for the clock to reset? And the last thing I'll show you is we have some preliminary results that suggest that for everything from a plus 12 shift to a minus 12 shift in the clock, we can show resetting properties on the order of a day or two, which would well outperform all the usual heuristics for recovering from jet lag. So we think we're beginning to get some insights at the organism scale.
Title of my presentation is, broadly speaking, Bioimage Informatics, and what I'm going to be talking about in the next 15-20 minutes are the various image processing, computer vision related activities, mostly in the context of bioimages, but also if time permits, I'll talk about some of the other interesting things that are happening, such as steganography. There are no slides on duplicate detection. Obviously, these days, detecting duplicate images from large-scale image databases is of interest in many different contexts. To begin with, I'll mostly focus on what we have been doing in the last 3 years or so within the Center for Bioimage Informatics, which is an NSF-funded center, an information technology research activity that has currently about 8 to 9 faculty and close to 20 plus other researchers, and has faculty in electrical engineering, in biology, chemical engineering, computer science, and statistics on this campus, as well as we have collaborations with Carnegie Mellon University and UC Berkeley. Okay, so this is a one-slide summary of the various information processing activities that's happening within the center. Again, broadly speaking, there are activities that are relating to large-scale image databases. When you have these collections of images, how do you manage them efficiently and effectively? We have ongoing activities on the pattern recognition and modeling side of the picture, primarily focusing on image processing, analysis, and machine learning tools to do data mining on those collections. And the focus in terms of the image collections themselves are primarily on, at the tissue and cell biology level, we have Professor Steve Fisher in the biology department, who is one of the leading experts on retinal detachment stuff. And so the vertebrate retina is one of the focus collections there. And then with Professors Stu Feinstein and Les Wilson, we are looking at videos of microtubules and various issues arising in that particular context, and I'll get into those details later on. This kind of summarizes, it's pretty difficult to see this close. The real problems that are there when you're dealing with these images. You have literally, this is just coming out of Steve Fisher's lab, for example. We have thousands of images that are being generated per project. And typically, each one of these experiments can result in gigabytes of data. And keep in mind that these are kind of small-scale experiments compared to the high-throughput imaging devices that are out there that can literally generate terabytes of data in an hour or so. And the real problems are that there are, the real issues here are that there aren't any efficient ways to organize, share, and search these images. And this is not just true for bioimages, but it is true for pretty much wherever you encounter images and video. Just think about your own personal image and video collections. What do you do at the end of the day? Maybe if you're lucky, you'll watch them once or see them on your computer once. But then they disappear, right? You don't know where they are. In fact, Steve made an interesting comment during our site visit time, is that sometimes negatives are much more easier to handle because you have cataloged them, you have put them in the cabinets, you can go and retrieve them. When they are on the computer, you don't know where they are. So you really need good tools to organize, share, and search these large collections. And that's pretty much the focus of the efforts in my lab for the past few years. The other way to present this is think of Google for images. And more than that, but at the base level, you would like to have a search engine that would take an input. An example input could be a query. A query would be like let's say I'm searching face images, and I provide my picture as the input query, and let the system go back and search and retrieve similar-looking faces from the database. So that's one example. Of course, in the context of bioimages, we are doing much more than search and retrieval. You may want to model the underlying processes and things like that. And as a first step in this direction, what we have, and this is something that's going to go online very soon, is a microscope to a database infrastructure pipeline that we have created. So the researchers, those who are actually imaging the stuff using microscopes can upload their images and the associated metadata information in an easy-to-use interface, which collects pretty much all of the information that goes with the images and then puts that into a database infrastructure. And the database part of it can do many of these functionalities that are outlined here. I may have some examples of some of the specific image processing tasks later on. For example, if I have an image like this, it came to me as a surprise that even a trivial task such as counting the number of cells in an image, I thought that's probably a solved problem. Apparently, there aren't any tools that are out there. For those of you who have worked with images and have used the NIH Image J software for looking at the images and doing some simple image processing operations, when I gave this presentation sometime back at Munich, one of the audience asked, so Image J has this cell counting tool, so what's new in this? So we went back and took a look at what Image J has. It has a very nice interface where you can move your mouse and click one at a time at each one of those cell locations, and it records those things. And it can record different types of clicks, meaning you can classify cells as cell A versus cell B, but that's what it has. Right now, it has a much better tool. We did complete this part of the project. So Image J has now the UCSB's automated cell counting tool. All you need to do is give the image, and it counts the number of cells in the image in an automated manner. The problem is not as trivial as it appears, by the way, because you need to do segmentation, you need to identify the region of interest, and there are lots of other processing complexities involved. And you can also do these kinds of stuff where you have the features that are extracted from these images, and you may want to mine those features to perhaps extract some interesting underlying phenomena. Much of the data on the retinal images coming from Steve Fisher's lab look like these images, where you have the cross-section of the retina tagged with different antibodies, and you are interested in understanding what's happening under these different experimental conditions. This is kind of a schematic of the problems, of one of the problems that they are studying. This is a normal retina where you have the various layers of the retinal cells outlined here. And if for whatever reason there is let's say there is a detachment, and that affects the various cells and layers over here, you can see that the photoreceptors are pretty much kind of degenerated, and then there are maybe other loss of connections and things like that. And you want to capture this in a quantitative manner for the biologists so that you can think about effective treatments later on. And talking about the retinal detachment problem itself, apparently it's a very common problem. One of the interesting things here is that the retina is, you can think of that as an extension of your brain in some sense, and it has some regenerative capabilities. And the retinal detachment can cause a permanent loss of vision if it is not treated within a certain span of time, typically maybe a day or so. And this is a picture that I showed earlier. So what are we interested in this particular exercise here? Let's assume that I have different samples of these images taken under many different experimental conditions. So the circles here represent normal images. Those here are, this is in some abstract feature space, okay? So these are 1 day after detachment. I have the features extracted from those images. These green dots represent 3 days after detachment, 7 days, 20 days, and so on. And hopefully they are well clustered, okay? So you have a nice kind of a progression going from the normal to the 28 days detachment. By the time you are here, probably you have lost most of your vision. So you probably want to fix this before you are at this stage. And what we are really interested in doing here is to be able to model this process, taking from the normal conditions all the way to the 28 days. Think of this as you have a knob to turn, and you can, by adjusting the parameters, you can see what's happening as you go through this process. And that's what we are currently looking at. And these are actually some real images coming out of the retina. This is the normal, 3 days after detached, 28 days after detached. And you can see the, I'm not sure if the contrast is good enough, but you can see the differences between the normal and the 3 days in the, so this is coming from this photoreceptor region over here. And obviously there are some visual changes, right? And right now, all that is done in a quantitative manner, in a qualitative manner, is that the biologist looks at these images and say, yeah, this looks different from this. That's about it. There's no underlying quantitative model for this whole process. Another example coming from somewhere over here, the ganglion cell layer. That's the normal 1 day detached, 3 day detached. And this is reattached after 3 days of detachment. So the retina is surgically reattached and then allowed to kind of grow for, well, whatever before the imaging was done, it was allowed to kind of continue whatever the reattachment process for 28 days, and then the snapshot was taken. And again, can you model the processes here? These are some of the preliminary results that we have, where we have computed the underlying image features and kind of shown this on a two-dimensional graph. And you can see some separation over here between the normal ones that are in the red. These are the green ones are the 1 day detached, the 3 day detached, and 3 days plus 28 days of reattachment. So there is some kind of a separation. We are trying to quantify this more precisely at this stage. Now, that kind of gives you the flavor of the bioimaging kind of problems we are looking at. But in terms of what are the underlying computer vision or image processing challenges, if I were to summarize this in one slide, these are the 3 critical problems in any image processing experiment. You need to segment, you need to establish the correspondence between the various image features, and then if you have the dynamic data, then tracking is an important problem. So what is segmentation? Segmentation is basically a partitioning of the image into homogeneous regions. For example, if I take the picture of this room over here, I would like to kind of segment out the various people from the background and the chairs and so on. So that's an example of segmentation. And we have really some good methods that are out there that can give the segmentation without any parameter tuning. Let me just get some water. More results coming out of non-bio images, and you can see the quality of the segmentation. Again, no parameter tuning, completely automated. More from the bio cases. So again, you can see the quality of the segmentation results. That's the original image. There are some contours that are outlined, and you can look at the quality of the segmentation results there. More of the same. So the bottom line is that we have good methods that are there that can do reliable and robust segmentation. I mentioned the cell counting problem earlier, and again, we have a really robust method that can count the cells at different granularities in the sense that these cells can come in different sizes depending on the layers that they are in. So it's kind of an adaptive system that can do the counting very robustly. And in terms of the accuracy of the cell counter, it's as good as what a human can do at this time based upon the preliminary evaluations that we have. I'm going to skip this slide over here. So moving on to the next problem, which is one of image correspondence. Correspondence is something that we do effortlessly, right? We have 2 eyes. At any given instant of time, you have 2 images that are being projected onto the retina, and we match those 2 images to get the perception of depth. But this is kind of an open problem in computer vision. We still don't know what the good computational model to achieve the correspondence and then put the things together. This is an example of putting together, building a mosaic coming out of 12 different slices of the retina, of retinal cross-section, and you can see the quality of the overall mosaic result. I'm not sure, again, given the contrast here, whether it's quite visible from the back. More examples of the same coming from retinal cross sections, and I do have some from other images. By the way, this is a fairly large mosaic that was created out of 40 images. There was a request from some group in Europe that wanted to mosaic this at sub-pixel resolution, and this is an example of a natural photograph where you can see the perspective distortions, right? These images are taken of objects at different depths, and this is a really difficult problem in terms of putting them together, blending them to give a photorealistic mosaic. If you have been on the campus, this is towards the engineering side, and again, few of those things put together in a seamless manner. It can do very well in cases where there are significant differences in the underlying image content. And here is an example of 2 aerial photographs that are taken 2 years apart. And you can see significant differences in terms of the image content, and the mosaicing algorithm still works really well in blending those things together. And this is a kind of a video mosaic that gives you the kind of, so you can see the motion, the underlying motion as well. And the real problems are when you have significant perspective distortions, when the objects are close to the camera. And this is a really cool picture that one of my students put together, and using the same ideas but in a different problem context. What we have here are images that are taken at different exposure settings, from somewhere that's close to zero aperture over time to something that's overexposed. And how do you blend this together so that you can get a really high dynamic range as far as the image is concerned? And the ones on the left and the right are the kind of the state-of-the-art results, and the one in the middle is what we can do today. And this is something that perhaps down the road you can put together, kind of integrate with a digital camera that can take a sequence of images and then blend the images to give really high dynamic range image. So that's about correspondence. Since I have 3 minutes left, let me outline one other problem and stop at that. So the third class of problems that one of the other problems that I mentioned before is the tracking. When you have motion in a video, tracking objects of interest is a very significant problem. And in the biology context, this is the kind of data that we are looking at coming from Les Wilson and Stu Feinstein's lab. This is a video of the microtubules growing and shortening. These are subcellular structures. And what they're interested in this particular problem is to actually identify those microtubules. I don't know whether you can see that. There are things that are growing and shortening, and track them, identify the microtubules, track them, record their growth and shortening, and then you can do some interesting statistical analysis following that. But the real challenge here is the first step, when you actually identify those individual microtubules and track them, and that's a very laborious process. And you can easily write a thesis just based upon analyzing a few hundred microtubules. And to cut the story short, we now have a fairly good mechanism to actually identify individual microtubules in large collections, track them in an automated manner, and I'm showing you some of the results over here. Not all the microtubules are being tracked in these slides, but you can do this reliably, and that's about 90% of the effort that goes into analyzing these kinds of data. And once you have the growth and shortening events, you can do a lot of other interesting stuff, including building models on top of this to kind of see how these microtubules react in a dynamic sense under different drug conditions and so on, and that's what the biologists are interested in studying. So again, repeating the slide that I had early on, the real issue here is that when you have large collections of images, you do not have good ways of organizing and sharing that piece of information, and that's pretty much what we are trying to do here at our center. So with that, I conclude my presentation. If you have any interest in some of the other topics, I'm going to be around here, so please feel free to contact me. Thank you.
So I'm going to tell you a little bit about this consortium we have that's aimed at developing tools for designing polymer formulations. And I'll tell you both a little bit about the model for the consortium and how it works, and also a little bit about the research that we're doing. This is basically a partnership between some academics, primarily here at UCSB, a set of companies, and some researchers at 2 national labs, Sandia National Labs and also at Los Alamos National Labs. And we're basically trying to develop computational tools, methods, and software for designing soft materials. And what I mean by that are alloys of polymers and also complex fluid formulations and solutions that would go, for example, into personal care products, shampoos, hairsprays, that sort of thing. And so we're interested in design tools and also understanding how these materials process, because the structure in the fluid and the structure in the material often depends on how you process the material, as well as the actual details, the molecular details of the formulation. So these are examples of the kinds of things we're interested in. We're interested in multi-component plastic materials, for example, that might go in both automotive exterior, rubber formulations and tires and things like that, interior plastics. You may not be aware, but many modern plastics are actually structured materials from kind of the 1 nanometer length scale on up to millimeter and larger scale, so spanning 8 to 10 orders of magnitude in structure within the material that ends up giving you the properties that are desirable in a given application. We're also interested in solution formulations, things like paints and coatings, which are actually very complicated mixtures of colloidal sized particles as well as polymers and other kinds of additives. And then if you go in the personal care arena, you pick up a bottle of a hairspray and you look at the list of ingredients, and it's just this mindless list of stuff that's been arrived at, a formula that's been arrived at by a formulation chemist sitting at a bench and mixing things. So what we're really trying to do is develop some software tools that will allow this design process to understand the relationship between the components that you put in the fluid, the structure that forms in the fluid, and then the ultimate properties of the fluid or the solid state that results from solidifying that fluid formulation. So with this consortium, what we're really trying to do is develop methods and models and schemes and ultimately software that we can share amongst the consortium, and that can be applied to address these kind of design problems. And we want to form a world-class center for doing this and to enhance interactions amongst all three types of partners and employment opportunities for the students and postdocs that are involved in this. So UCSP is the focal point for this consortium. And the different kinds of partners contribute obviously different things. For the financial support is primarily coming from the industrial partners. And there's kind of two levels of partnership. There's a sort of 35K level and then a full level. And a full partner will typically support a graduate student or a postdoc in entirety. And the project will be designed in a way that we will contribute tools and know-how for simulating a broader class of systems than we already know how to describe and simulate. But also we'll kind of custom tailor that project to the specific interests of the company that's providing the support. One of the primary outcomes of this consortium is software. And one reason this consortium works, even though some of the partners in the consortium are competitors out in the marketplace, is that we're providing software tools they can take inside the company and use in a proprietary way in designing and developing their own products. So the basic deal is if you're part of the consortium, then you get access both to the source code and the executables, and you can use that internally for your own purposes. So these are the faculty who are involved here at UCSB, and we're coming from some different departments. We've got a couple of young faculty in the math department who are very good at the types of numerical analysis that are involved in developing these models, and then Sanjoy Banerjee and myself from chemical engineering. And we've got some colleagues at Los Alamos and Sandia who also have complementary modeling skills who are quite involved in developing these tools. So these are our current partners. We have two of the largest chemical and materials companies in France, Arkema and Rhodia, largest chemical company in Japan, Mitsubishi Chemical. General Electric has been part of the consortium, Dow Chemical. Nestle Research Center, they're interested in polymers you can eat that go into chocolate, things like that. Kraton Polymers in Houston was formerly a division of Shell Chemicals. So we're doing a lot with what are called block and graph copolymers. Many interesting modern polymer formulations, especially that relate to nanotechnology, involve these kinds of molecules. They're basically segmented polymers that have, for example, in this case, a polystyrene sequence and then a different kind of polymer sequence, for example, polyisoprene or polybutadiene. This is a kind of classical way of making what's called a thermoplastic elastomer, material that will stretch and recover, but you can heat it up and it will flow. So you can make things like tennis shoe soles and adhesives. But what's exquisite about these kinds of molecules is they can form very intricate nanostructures just by self-assembly down at the 10 nanometer scale, which you can control by controlling the architecture of the polymer, how you sequence the blocks and how long they are, which you can control at the time you do the chemistry. You can get all kinds of different structures, and the materials properties will depend on those structures. There's a lot going on in the synthetic chemistry world, which has enabled the ability to create lots of different interesting polymers from readily available, inexpensive building blocks, things like vinyl chloride, ethylene, propylene. Very inexpensive building blocks that can be used to build polymers are now, with these new chemistries, you can build them into these block and graph forms. So you can get a much broader range of polymeric materials into this space of being able to assemble these very detailed kinds of nanostructures. So the tools we're actually developing in this consortium we refer to as field-based computer simulation tools. Most molecular simulations of fluids are done with atomistic models. So you build site-specific models for different atoms in your large molecules, and you postulate force fields for how those atoms interact. And the problem with that approach is basically you can simulate with even a pretty large computer cluster these days up to about 1 million atoms for about 10 nanoseconds of real time. All right? And these kinds of fluids will typically have relaxation times that are seconds. So 8 to 10 orders of magnitude longer than what you can get at by a kind of atomistic bottom-up modeling approach. So the time scales are way off. The structural scales, it turns out, are way off. All right? So the approach we're taking, and these are, for example, TEM images of the kinds of fluids we're interested in. We're interested in fluids that have structure from kind of 1 nanometer length scale, possibly up to microns and millimeters. So we've got to bridge many orders of magnitude in scale. And so what we're actually doing is building models based on describing the structure in the fluid based on continuous fields rather than individual atoms. All right? And there's a formal kind of mechanism for doing this. I'm not going to go into the theory of it, but actually you can take any kind of atomistic or even coarse-grained model of an atomic or a molecular fluid, and you can formally convert it into what's called a statistical field theory, where the degrees of freedom in the fluid, we call them these W fields, are basically fluctuating chemical potential fields. And so if the chemical potential for species A is higher here, there's more atoms of A over here versus atoms of B somewhere else. And the beauty of computing with fields is we have a lot of good numerical methods, for example, in computational fluid mechanics and solid mechanics, in density functional theory for electronic structure. We know how to resolve fields by lots of different methods to sort of resolve the structures that you care about. So you simply resolve the fields to whatever scale you're interested. So examples of the kinds of things we can do with this, we can, for example, if you have these periodic structures, we can zoom in and do very high resolution simulations of one periodic unit cell of the structure and calculate the free energy of it very accurately. We can also do what we call large cell simulations. This is actually a simulation relevant to using these block copolymer materials in thin films to do submicron lithography. And here what we're trying to do is a kind of combination of a top-down and a bottom-up approach. Use a top-down with conventional lithography to put in this case, a pentagonal well into a silicon wafer. You pour your block copolymer in, and you allow the self-assembly to occur within the well. And so what we're interested in is how these edges can perfect the order of self-assembly near the edge. And obviously, if you have a system that wants to form locally a hexagonal lattice, you put it in a pentagonal cell and it's going to be frustrated. So here you get lots of defects. But if you change that well to a hexagon, it turns out every time you lay a film in there, you'll get a perfect structure if you've got the length of the sides of the hexagon commensurate with multiples of the kind of fundamental lattice spacing. So this is a way you can, in principle, with a combination of top-down lithography and self-assembly, get perfect registry of these 10 nanometer domains from one side of a wafer to another. So we can do these kind of unit cell calculations. That's an example of a unit cell. We can do formulation problems. For example, this little project we did with Arkema. Arkema makes plexiglass, so sheet acrylic resin, and they're interested in making it tougher, more resistant to impact, but still transparent. And so the strategy is put a little bit of one of these block copolymer additives into your plexiglass, and the idea is to have this little red block, which is dark in these images, to be a rubbery domain, but down at the nanometer scale that doesn't scatter light. Okay, and so this is just a whole palette of simulations that we run, and you kind of want to be in this space where you have these very small, fine domains well below the wavelength of light that don't scatter light. And you can zoom in. This is an example of a kind of high-resolution simulation, and the point is we're accessing TEM relevant, microscopy relevant length scales with these techniques that you could never do with a kind of atomistic-based simulation technique. Okay, so here's an example of this this graphoepitaxy business. If you do the self-assembly inside a hexagonal cell as opposed to a pentagon-shaped cell, if the side of the length of the side is just about right, every time you get a perfect lattice in between, whereas if it's too small, you can see some defects there, or if it's too big, you get defects. So we're using this in actually designing some lithography strategies. We've also learned in some of these projects how to combine tools for simulating rigid inorganic nanoparticles within these complex fluid formulations. So there's a lot of interest these days with the much enhanced availability of a whole wide variety of inorganic nanoparticles, both metal nanoparticles, semiconductor nanoparticles, combining these and dispersing these in polymer matrices for a whole wide variety of applications. And so you'd like to have techniques where you could do simulations of the coupling between the structure of the complex fluid and the location of the particles in the dispersion, the quality of the dispersion. So here's an example where we have both experiments and simulations of nanoparticles that are dispersed in one of these block copolymers. In this case, it's a lamellar type structure. You put the particles in at low concentration, and if they go into the light region, you increase the concentration of particles, they will actually drive the fluid into a different structure. So here's a case where there's an interplay between the structured fluid controlling where the particles go in the dispersion, but also the presence of the particle changing the structure of the fluid in a complicated way. Okay, so that's kind of what the CFDC is about, and I've given you a few examples of the kinds of things we're doing. So, you know, this structure has really enabled us to develop both methods and software. And we're applying these methods to systems that are kind of collective interest to all the partners in the consortium. And these are kind of the emerging areas that we're working in. We're working very hard in this area of nanoparticles and colloids combined with structured fluids. Many formulations, especially in the personal care arena, are done in water. And many of the polymers that dissolve in water have charges on them, and they're polyelectrolytes. And so this is a very interesting area of formulation space, because as soon as you put the polymer into water, you get dissociation, you get charges, and you have electrostatics that play in a very complicated way with these self-assembly processes. So we're developing tools to work in this area. It's very easy to get complexation and things falling out of solution. Another interesting emerging area is what's referred to as supramolecular polymers. And this is built around a sort of modern theme in chemistry of supramolecular chemistry and supramolecular assembly, basically using non-covalent bonds, so bonds that have intermediate strength, like one hydrogen bond or two hydrogen bonds or three hydrogen bonds, that sort of strength, to put together large structures that are thermally reversible. So you can imagine a polymer system that at low temperature all these bonds are in place and it acts like a cross-linked rigid material with good mechanical properties. You heat it up, all these bonds break, and the material becomes very fluid and easy to process. So you can get very interesting combinations of properties by putting these reversible bonds into a system in very specific ways. So there's a lot of interest and activity in the experimental world in this area, and we think that having some modeling and simulation tools to help guide experimentation in this area is useful. And then finally, there are very, very few tools for understanding how these kinds of structured fluids process. All right? And many times, and particularly in the plastics world, the structure you get in the material depends very much on how you process it. So whether you blew a film to make some kind of a sheet material, you extruded something, you injection molded it, you can get very different kinds of structure in the material, and you'd like to be able to understand the relationship between the processing history, the fluid mechanics, and the structure. So we're trying to develop tools in that space. And finally, it's a fun area to work, just in principle, because we're bringing together tools and methods from lots of different fields. So the theoretical formalism has a lot of similarities to what people do in branches of theoretical physics like quantum field theory and high energy physics. We're using sort of state-of-the-art numerical methods and computational tools, and we're doing calculations that are relevant to real material science and real-world kinds of formulations.